Hello students, in previous part we have discussed about transcription process. In this topic we will discuss on genetic code. Genetic code. As we have in earlier part we discussed about central dogma. Central dogma is a process in which DNA converts into mRNA and then mRNA to protein. This is the central dogma in which the information can be sent out. The process in which DNA changes into mRNA, that is called transcription. About this process we have already discussed in previous part. Translation means RNA mRNA changes into protein, that is called translation. So the information transfer from in any organism through this process. Protein. Before going to protein, we will discuss on genetic code. We know that there are 4 nitrogen bases present in DNA. 4 nitrogen bases and 20 types of amino acid present. How the genetic code may be arrived? Because DNA and RNA both act as genetic material, in which we can say that these are made up of 4 nitrogen bases and 20 types of amino acid. DNA and RNA both are the genetic material which are expressed from 4 nitrogen bases or nitrogen bases that may be made up of 20 types of amino acid. How these amino acids represent the genetic codes? As we have already discussed, amino acid always read in triplet. It means we always read in triplets, such like as Ram has big cat. It means always there are 3 words in the one sequence of amino acid. We always read in triplet. So there is the riddle for many scientists, many biochemists, physicists and biologists, how these genetic code may be expressed. It was George Gamow. George Gamow, who represent that there are 4 nitrogen bases with 20 types of amino acid makes the 64 code. He proposed that there are 64 codes represent 20 types of amino acid which contain 4 nitrogen bases. With the help of permutation combination, it means always the amino acid read in triplet and there are 4 nitrogen bases, so 4 into 4 into 4, that is equal to 64 codes present which represent the 20 types of amino acid. This was proposed by George Gamow, genetic code proposed by George Gamow, and George Gamow was quoted by some other scientists also. Those are Dr. Hargobind Khurana, Marshall Nirenberg, and Severo Oka. These were the 3 scientists who supported the genetic code methods of George Gamow. How these 64 codes are represented? As we have discussed that in nitrogen base, mRNA or RNA contain 4 nitrogen bases. 4 nitrogen bases in RNA. U, A, C, G. In place of thymine, because in DNA thymine present, but in RNA this thymine is replaced by uracil. How these 64 codes are represent. Firstly, we discuss with the table. We'll make one or two slot. This is the first site from where we will read out the first sequence U, A, C, and G. This is the first side. The another side is here, this one. U, A, C, and G. Same sequence. And they are present in triplet. So third side we will here write it here. U, A, C, G, U, A, C, G. How these codes are designated or produced? We'll make one or two slots. First we will take the first U, and from second U, and third this first one is the first sequence. Second one U, and second letter of third sequence A, U, A. Third one is U, C. And the fourth one is U, G. So in one slot there are 4 generic codes, and there are 16 slots. 16 slots, total 16 slots, and one slot contains 4 amino acid, 4 generic codes. So we can say that 16 into 4, 64. George Gamow represented that there are 64 codes present. How these codes are arranged? Just one slot, again one slot, U, A, U. Again one is U, first U, secondly here is A, and this one is U. Again U, A, U, A, C, U, A, G. Same. Next one U, C, U, C, A. U, C, C, U, C, G. We will write it. There are 64 codes present. This was proposed by George Gamow. And not only George Gamow, he was supported by 3 other scientists, Dr. Hargobind Khurana, Marcel Nirenberg, and Severo Oka. These are the 64 codes. Next, what are the salient features of 64 codes? Salient feature of generic code. The first one is there are 64 codes present. There are 64 generic codes which represent the amino acid. There are 64 codes or 64 generic codes present. Out of, or you can say that only the 61 codes represent the amino acid. It means there are total 64 codes, and out of 64 only 61 codes represent amino acid. The rest 3 which do not represent any amino acid, so those codes are known as nonsense or stop codon. And 3 codes which do not represent any amino acid. So these 3 codes are known as a stop codon or nonsense codon. Therefore, also these 3 codes are known as nonsense codon as well as stop codon. As we draw the diagram or table, in the table we have seen that there are 64 code. Out of 64 code, only 61 represent any amino acid, but 3 genetic code do not represent any of the amino acid sequence. So these 3 are known as nonsense as well as stop code. This is the first salient feature. Second one is each genetic code represent only one type of amino acid. Each genetic code represent only one type of amino acid, not other. So we can say that there is an ambiguous means no confusion that one amino acid represent this one or that one. No, only one type of genetic code represent only one amino acid, and this is called one type of amino acid. So it is unambiguous or non-ambiguity means no confusion. There is no any types of confusion which amino acid represent this one that or that one. Only one type of genetic code represent one amino acid. The third one is genetic code genetic code representation never use punctuation. It means no use of comma and full stop, dash, etc. No punctuation. Is used in amino acid sequence. So it's like as U A C U A G. We don't write any commas, don't use any commas, full stop and etc. It is always written same. No gap. No, this one like again U A C. There is no mention of gap. No punctuation, no gap, or no gap is used. Number 4. There is no any overlapping during representation of an amino acid sequence. No overlapping in amino acid sequence. For example, if we take an example of UAG. This is the sequence. We always write in first reading triplet. It means this one line. Triplet. No overlapping. Somehow, firstly we will discuss UCU. And again we remove U, then makes the CUU. No overlapping can be take place. It means we only once read out only one sequence. No overlapping in amino acid sequence, and there is no other. It means never repeat any sequence of amino acid, and it always read out in triplet. So there is always reading triplet. And the fifth one is the genetic code sequence is universal. It means it is accepted by the universe. Genetic code is universally accepted. For example, if we take the example of UUU, whether the organism is prokaryote or other, means higher. The triple UUU always represent only one type of amino acid, that is phenylalanine. It means there is no any ambiguity, there is no confusion. One generic code represent two or more. No, there is no confusion. And sixth one is only one type. We have written that each generic code represents only one type of amino acid. But one exceptional case, AUG. AUG is the generic code which function dual. It acts as methionine amino acid as well as start codon. AUG acts as methionine or start codon. So and start codon. So it performs dual function. It performs dual function. Exception case, one more is GUG. That is also act as start codon as well as valine. One is AUG. Mostly organism AUG act as start codon where the mRNA change into protein. That site from where the mRNA change start to change into protein, that site is known as a start codon, initiator codon. So AUG act as a methionine as well as a start codon. One more is GUG. That is also act as in some organism start codon and valine. And we have discussed that how to 64. 61 are represent any amino acid. The three codes which do not represent any amino acid, those are called stop codon, and those three stop codons are the three stop codons are UAA, UAG and UGA. UAA, it is known as occur. UAG, it is known as amber, and UGA, occur. These three codes which do not represent any amino acid, they act as stop codon. So they are known as nonsense or stop codons. Clear? This is the topic in which we have discussed how the generic code is represented. What is the role of generic code? Because we know that as the mRNA changes to protein, the protein amino acid sequence is, there are 20 types of amino acid, in which 4 nitrogen base present, but how these arrangement may be occurred. This was the riddle for many scientists, but at last George Gamow represented by the permutation combination 4 nitrogen base always read out in triplet, which represent 20 types of amino acid. Permutation combination is 4 into 4 into 4, that is 64. So he showed that there are 64 total codes of amino acid, which represent 20 types of amino acid. This discovery was supported by 3 more scientists, Dr. Hargobind Khurana, Marshall Nierenberg and Severo Oka. How these codes are generated, we have seen this one like. Then what are the selling features occurred, or what are the selling features of generic code? In next part we will discuss on translation.